Comparative anatomy requires us to take a look at different kinds of organisms, dissect their structure, how they are put together, and find similarities between two different species. So, for instance, one thing that we might look for is called a homology. Homology. They have the same homology, logic to them, logos, right? You have the same idea behind them, a homology. These are structures that are similar to each other because they are evolutionarily related to each other, because they both descended from the same ancestral structure. Maybe they've been modified for different purposes, so these structures might look somewhat different from each other, but if you look at their internal structure, they will be very fundamentally similar. For example, the bones in a human arm have a very particular arrangement to them. You have a long bone, which is called your humerus. You have two smaller bones, called your radius and your ulna, which are thinner and can actually rotate around each other, giving you a nice range of motion. The little tiny bones inside of your wrist. You have your metacarpals, your digit bones, which help you grab things. Now, this arrangement is really, really nice for a human arm. It's really well suited to climbing, to grabbing things, to holding tools, to manipulating objects, to expressing yourself while you're talking. The human arm and hand is a highly evolved organ, and it's extremely well suited to its particular purpose. However, that same arrangement of bones shows up in other organisms as well. If you take a look at this cat's leg right here, the foreleg of a cat, you can see that same order of bones. You have the long bone that's connected to the shoulder, the single bone. Then you have the two smaller bones, which have a little bit more flexibility to them. They've been arranged in a slightly different way to make them good for bearing weight, but you can see that it's the same kind of bone structure. Then you have all those tiny little bones right down there at the paw, and then the longer digit bones. The exact same bones, a little bit of different shape, but the exact same kinds of bones in the exact same positions. And the cat's paw is also an extremely highly evolved organ for the purpose that it's used for. It's used for being agile and bearing weight and walking around and moving and clawing at prey and things like that. So the cat's paw is also a highly evolved organ using the same arrangement of bones. The shape has changed, the specifics have changed, but the same idea is there, the same logic is there. Homology, the same logic to it. Next, you can take a look at a whale's flipper. Now, they have the same order of bones. One long bone attached to the shoulder, two smaller bones, radius and ulna. You have a series of very tiny bones, and then you have the digits. But wow, do they have a lot of digits there. As it turns out, that's actually a pretty easy mutation to cause. There's a certain kind of gene called a homeotic gene. And homeotic genes don't code just for a specific protein, like all the other genes we've talked about. They're more big picture. They're more about the overall design of the organism. So it'll say something like, put digit bone here. And if you get a mutation which causes a gene duplication for the gene that says put digit bone here, you can get multiple digits. We see this sometimes with humans where they have a mutation which causes them to gain an entire extra finger, right? You can get these kind of large-scale changes with relatively simple mutations. So this is what's happened with the whale. Now you might also notice that the whale's bones are short and thick compared to, say, the human arm bones. But, as it turns out, changes to tiny little proteins that guide the development of an organism can alter that. They can say, okay, grow the bone this long and stop. They can say, grow the bone this thick and stop. So these types of mutations are going to allow organisms to adapt their particular body parts, their organs, to the environment. Populations evolve to adapt to their specific environment. What's the whale flipper adapted for? It's adapted for gliding through the water. So these extra digit bones give it a lot of flexibility to allow it to dexterously glide itself through the water. Same idea, take a look at a bat's wing. These are all mammals, relatively closely related organisms, and you can see that the bat's wing actually has the same bone structure as all of these other mammals' forelimbs as well. One long bone, two thinner bones, and then you have the digits coming out. Now, these are all very thin and elongated, but again, very tiny tweaks to proteins can cause it to become thin and elongated. And this is modified for flight. If you take a look at a bat's wing and you use your imagination, you can actually see that it's kind of like a creepy human hand with extra long fingers sticking out in order to allow their membranous wing to have as much surface area as possible when they catch the air and are able to fly around. So these are homologies. These are organs in animals that are similar to each other in construction because they share an evolutionary ancestor. They might be modified for different purposes, but they have the same construction to them, so they're related. Another aspect of comparative anatomy is not just finding where things are closely related, but figuring out which comparisons are actually less important. For example, if you take a look at the wings of an insect and the wings of a bird, they're both there for flight. But if you actually look at the wings of a bird, you'll find that it has a very different anatomical construction from the wings of an insect. The wings of a bird have bones inside of them, thin hollow bones in order to lighten up the load, in order to allow them to fly easier, but bones. In fact, it has the same bone structure as all of the organisms on that previous slide. Now, some of the bones have been fused together, some of the digit bones, in order to create this wing structure, but overall, it's a very similar setup of bones. A little bit more distantly related than the others, but in that same category. And those bones are articulated by muscles which go all the way throughout the wing, and it's able to catch the air by virtue of these feathers that it has. That's what a bird uses in order to fly around. Insect wings, on the other hand, are supported by a structural polysaccharide which we call chitin. So there's no feathers involved there, and also insects don't have any bones to speak of, right? It's just that exoskeleton, that chitin, that's holding that dragonfly wing together. Now they do have muscles, like the bird has muscles, but the bird has muscles going all the way through that structure of the wing, whereas the insects, all of their muscles are kind of concentrated on their back, right? And they have to articulate them in a slightly different way. Although these organs have a similar purpose to them, they're both there for flight, they clearly have a fundamentally different construction to them. The wings of an insect are not similar to the wings of a bird, and the wings of a bird are only superficially similar to the wings of a bat. These three organs all developed at different points in evolutionary history, so they are analogous. This is what we call convergent evolution. Some features are just so fundamentally useful that multiple different lineages independently develop them at different points in time. So insects learned how to fly, birds learned how to fly, mammals learned how to fly. If you take a look at things like pterodactyls, we know that more lizard-like creatures learned how to fly. We have flying fish. They can't actually fly, but they can glide a good distance. Flight turns out to be such a handy evolutionary trick that many different branches of the family tree came up with the same idea at different points in history. Well, when you have organs which are similar to each other because of convergent evolution, but are not actually related to each other, they showed up separately in different lineages, we call that an analogy. An analogy. In English, just in the English language, when you make an analogy between things, you're drawing a comparison between two dissimilar ideas. We're using it in a similar sense here. An analogy is a comparison, saying how these things are similar between two unrelated structures, okay? So analogies are not related by evolution. Homologies are related by evolution. Another concept that we could talk about in comparative anatomy is the idea of a vestigial structure. There are some good examples of vestigial structures in humans. For example, goosebumps, right? You get scared, you get goosebumps. Your hair on your arm stands up on end, right? Or if you get cold, your body springs up with goosebumps. And those goosebumps don't really do anything for you. They're not even remotely helpful to a human person. However, if you look at organisms that are closely related to humans, other kinds of mammals or even birds actually, you will see that the goosebumps do have a kind of purpose to them. If, say, a cat gets scared by a predator, what does it do? Its hair stands up on end and it makes the cat look bigger and more intimidating, so it helps scare scare off that predator a little bit. So the goosebumps are helpful in that case. If a mammal gets very cold, its hair will stand up on end, which will increase the amount of air which is trapped near that mammal's warm body. Their body will warm up the air and give us a nice insulating layer in order to help stave off the cold. Now, humans, through evolutionary time, we've lost a good amount of our body hair, but we still get goosebumps, even though there's no benefit to it. And the reason is because having goosebumps doesn't have any kind of disadvantage associated with it. If it was actively harmful to our survival and reproduction, then evolution would have weeded goosebumps out a while ago. But there's really no problem with goosebumps. You get them, and it's kind of just a quirk of human anatomy, but they don't really do anything for or against you. Another vestigial structure that you find in humans is the tailbone. Right? This little nub of material at the very, very base of your spine here doesn't serve much of a purpose, does it? Except getting broken periodically. I'm wagering that some of you have landed too hard on your tailbone, you've broken, and it's been very, very painful. But it doesn't really do anything for us, this tailbone. However, organisms that are closely related to humans have tails. So monkeys, for instance, are a kind of primate, and primates are the order of mammals that humans are also classified into. We're a member of the great ape family, but monkeys are in the primate order along with us, and we're all kinds of mammals. What this tells us is that at some point way back in evolutionary time, some pre-human ancestor probably did have a tail. And there's actually good evidence for that as well in living humans. Periodically, a human will be born with a tail that can even move around a little bit. Now, that tail is usually surgically removed at birth, but it is nonetheless there. We have the genes inside of our genome for growing a tail, just periodically they get switched back on. Genes which have been turned off that serve some sort of function in an ancestral form but are now just not active, they're a kind of vestigial trait. We call them atavistic genes, atavistic traits, atavisms, the kind of genes that are just switched off. And there's lots of atavisms inside our genetic code. If you wanted to take a look at vestigial organs in different kinds of organisms, then you could take a look at snakes as a pretty good example. Snakes are descendant from organisms that had legs. They're kind of related to lizards and other reptiles, and they all have legs. So snakes, ancestors of snakes, used to have legs. Over time, they lost the need for those legs. They found it more useful to be able to burrow into holes, fit into tight spaces, be able to constrict around things, and legs were just kind of getting in their way in that process. But if you look very closely at the skeletons of certain snakes, you will still find the remnants of those legs. For example, these little spikes here are bony protrusions, and they are the vestigial remnants of legs. In whales, we see remnants of back legs, hind legs, still inside of their skeleton, internal to their body, but nonetheless there. But vestigial traits aren't eliminated from the species because there's no survival disadvantage. There's no reproductive disadvantage associated with having these traits. Related to comparative anatomy, we have comparative embryology. In comparative embryology, you do the same kinds of checks between organisms, but you're looking at earlier developmental stages. It's an interesting fact that all vertebrate embryos, everything with a backbone, with a spinal column, look very, very similar at early stages of development. I mean, we all start as a single fertilized cell, but the differences between us don't start to show up visibly until later stages of development. If you look at a couple weeks of development in, mammals and birds and fish look somewhat superficially similar to each other. There are some differences starting to show up, but there are a lot of similarities here. Now, one thing you might notice is, hey, look at that bird embryo there in the center. It's got a long, bony tail. And you think, well, yeah, birds have tails, don't they? But no, they do not. Birds have tail feathers. They don't have a long, bony tail coming out the backside. And yet this bird embryo seems to have a long, bony, lizard-like tail coming out the back. What's the deal with that? Well, at a certain stage of development, an enzyme starts to break down that tail, that bony tail, and it's not seen in the adult organism. So this is an example of the value of comparative embryology. We would never have noticed that kind of long, bony, lizard-like tail in birds unless we had checked their embryos. There's also a structure on all three of these embryos which we refer to as pharyngeal pouches. So all of these embryos kind of look like this, right? You have this head, and kind of goes around like that. You got these little nubs, which will become limbs later on. You have this kind of big eye spot. And you have these like pouches underneath. Those pouches show up in all three kinds of embryos. Now, interestingly, in humans, that pouch will develop into part of our inner ear system. In fish, it will develop into a portion of their gills. So these two organs, the human inner ear structure and part of the throat, and fish's gills, are actually related to each other. They develop from the same early structure, and that's something we may not have noticed if we were only looking at adult organisms. So this is comparative embryology, looking for yet more homologies between organisms by checking their earliest stages of development before they kind of differentiate into different forms.